Assalamualaikum. A warm welcome to all of you, ladies and gentlemen, on our first International Brain and Biomedicine Conference, IBBC 2023. I'm Irsa Javed, MPhil scholar from Department of Physiology, Government College University, Faisalabad. I'm going to moderate this session. This session includes different talks from various fields of sciences, such as toxicology, pharmacy, and environmental science. So let's make a start of today's session. Our first presenter is Dr. Shabnur Iqbal from Government College University, Faisalabad. She will present her research work on the title of Sparosine mitigates valproic acid induced reproductive damage via modulating PI3K AKT mTOR pathway. Please, Dr. Shabnur Iqbal.
Mitigates sulfuric acid-induced reproductive damage by moderating P13K AKT mTOR pathway. Sulfuric acid is low-cost drug and is used to treat various diseases, and its long-term use cause endocrine impairment, particularly in reproductive system, and alter the hormone profile, and also reduce the sperm count and quality and testicular atrophy that led to male infertility. Hence, our study used the Sabrosan to treat the valproic acid-induced reproductive damage, that is a classical recombinant derivative that exerts notable antioxidant and anti-inflammatory properties. Aims and objectives: investigate the reproductive effect of the Sabrosan against valproic acid-induced oxidative stress, inflammatory cytokines, and change in male hormone profile in male rats, and to investigate role of Sabrosan in modulation of AKT-P13K and MTR pathway methodology. Valproic acid is dose is selected from previously reported study, while for the Sabrosan we conduct the toxicity test and observe no mortality up to 2000 milligram per kg. Hence, Sabrosan was safe to use and selected we select two doses 50 and 100 milligram per kg to assess their reproductive effect. Experimental design: the rats were divided into four groups, and the valproic acid and Sabrosan were co-treated for 28 days and given orally, and compared with the rats who were only treated with the valproic acid and control groups. And each group contains the five rats. Biochemical analysis that include ELISA, real-time PCR, uh, for to quantify the messenger RNA expression and histology. These are the primary sequence for target uh, genes for RT-PCR results. The result of the oxidative stress assay shows that the uh, well, shows that the after the treatment of the Sabrosan, glutathione peroxidase and catalase were significantly increased as compared to the rats who were only treated with the valproic acid. ELISA male hormone profile uh, shows that the TSH, follicle stimulating hormone, and luteinizing hormone were significantly increased after the co-treatment of the Sabrosan and valproic acid as compared to the rat who were only treated with the valproic acid. ELISA for inflammatory cytokines shows that the inflammatory cytokines were significantly reduced after the co-treatment of Sabrosan and valproic acid of as compared to the rats who were only treated with the valproic acid. Result of the RT-PCR shows that the AKT, P13K, and MTR messenger RNA expression were significantly increased after the co-treatments of the Sabrosan and valproic acid as compared to the rats who were only treated with the valproic acid. Results of the histopathological analysis shows that the rats who were only treated with the valproic acid, whose seminiferous tubules were damaged, and observed less primary and secondary spermatocytes, and also observed necrosis around the seminiferous tubules and cloudy swellings. While after the co-treatment of the valproic acid and Sabrosan, we observed the improvement and of the seminiferous tubules and observed increased number of the secondary and primary spermatocyte and their structure is improved, and also the desk load is swallowing as compared to the rats who were only treated with the valproic acids. Conclusion. Suppressant reduced the oxidative stress and inflammatory cytokines that were the key elements to inhibit the AKT-P13K MTR pathway. Suppressant upregulated the AKT-P13K and MTR pathway that help to proliferate spermatogonia to increase the number of primary and secondary spermatocytes. Moderation of the key events assisted to improve FSH, LH, and TSH to instigate centralized cells and leading cells to promote spermatogenesis. Suppression was exactly dose-dependent effect, and suppression might cause uh, suppression might used as a co-supplement with valproic acid medication to avoid its side effects after the proper clinical trials. Thank you.
Thank you, Dr. Shabnur Iqbal, for such an appreciable talk. Our next presenter is Ikra Chaudhary from Quaid-e-Azam University. The title of her presentation is Strategic Role of Microneedles and Nanotechnology in Brain-Related Diseases. Please, Ikra Chaudhary from Quaid-e-Azam University.
Assalamu alaikum. We are here to discuss brain health, a matter of life and a prosperity for a developing nation in the International Brain and Biomedicine Conference organized by Department of Physiology, Government College University, Faisalabad and sponsored by International Brain Research Organization and Dana Foundation. Me, Ikra Chaudhary, here to present strategic role of nanotechnology and microneedles in brain-related diseases. I am a PhD scholar in Pharmaceutics, Department of Pharmacy, Quaid-e-Azam University, Islamabad. Let's start with a brief introduction of major structural parts of the brain, such as parietal lobe, occipital lobe, frontal lobe, and temporal lobe, cerebellum and spinal cord, and brainstem. The CNS diseases and disorders are posing a great challenge for mankind. Today, neurological disorders are among the highest reasons for causing death and disability in the world. Neuropsychiatric disorders and neurodegenerative are two major divisions brain-related disorders. The neuropsychiatric disorder involves the unusual thoughts, perception, emotions, and disturbance in behavior, and in the subset in absence in mental disorders. And the neurodegenerative disorder in which nervous system stops working and or die and usually gets worse over time and have no cure. The neurodegenerative diseases are Parkinson's diseases, Alzheimer, Huntington disease, multiple sclerosis, spinal muscular atrophy, and spinocerebellar ataxia. And the neuropsychiatric diseases are seizures, cognitive deficiency disorders, uncontrolled anger, migraine, epilepsy, addiction, and eating disorders. The wide spectrum of potential drugs are available to treat these neurological disorders, but still there is a gap in the therapeutic success due to the different range of challenges. One of the most common challenges in CNS related disorders are the encountering in the peripheral barriers. Peripheral barriers are the blood-brain barrier in which tiny junctions are present across the brain, which unenable the delivery of different type of drugs or entities or substances across the brain, and limited the entry of many drugs via blood-brain barrier due to their insufficient entry in brain, as well as a limited access to immune cells. The most of the drugs used to treat these brain-related disorders are lipophilic in nature and having 400 to 500 Dalton molecular weight, and are unable to cross blood-brain barrier and in its pharmacologically significant quantity. So that's why there is a need of that technology to deliver these drugs across the blood-brain barrier in pharmacologically significant quantity. We utilize nanocarriers of particle size between 1 and 100 nanometer have been emerged by the development of nanotechnology, among which polymeric nanoparticles, solid-lipid nanoparticles, liposomes, and micelles made their debut as nanocarriers for the treatment of various neurological disorders. But nowadays, newer and more advanced nanosystems such as dendrimers, nanoemulsions, nanogels, nanosuspensions, nanotube, etc., which have shown great potential than the previous drug delivery systems. These nanoparticles have optimum size, surface area, surface charge, and should be biodegradable, non-toxic, biocompatible, cost-effective, and side-effective. These these nanoparticles encounter the blood-brain barrier with different passage of insertion. Means transcellular cytosis is utilized to deliver drug across the different substances across the blood-brain barrier, in which these substances move through the apical or basolateral membrane, and the other mechanism is the paracellular transport, in which epithelial membrane utilized to deliver substances across the blood-brain barrier. The other is the flux transport, in which different type of B glycoproteins are utilized to deliver hormones, waste, toxins, and remove or move inside the brain. And the receptor mediated interactions were used by the budding of different hormones, different genetic materials, different drugs inside the brain by a budding mechanism. And the other one is a carrier mediated transport, in which different carriers are present on the cell blood brain barrier cell membrane, such as glucose carriers, which utilized to deliver glucose across the brain. Because glucose is not formulated inside the brain utilizes glucose from the outsource. And other is the absorptive transcytosis, in which different electrostatic interactions was utilized to deliver drugs or any other substances across the blood-brain barrier. And blood-brain barrier is a negatively charged moieties and used to tackle positively charged drugs inside the brain. The nanoparticles deliver drug by using these mechanisms and moved towards the blood-brain barrier and delivered the drug inside the brain. The nanoparticles are basically divided on the basis of materials. Mean different type of materials were utilized to formulate these nanoparticles, such as lipids, polymerics, carbon nanotubes, metal particles, etc. And the size differentiation was reached was 100 nanometer to 1 nanometer, and different surface substances are attached on the nanoparticles, means different type of pegylated or other coatings. Surface functional groups are attached, means thiolation, or different type of carboxyl groups are attached to nanoparticles to tackle the receptor mediated or carrier mediated transport. Different type of surface charges was developed or optimized across the cell across the nanoparticles, and different targeted ligands was attached with nanoparticles. And on the basis of types, on the basis of shape, different nanoparticles are formulated, which are of spherical, cube, rod, or star, or plate-like structures. And these nanoparticles utilized to deliver the drug across the blood-brain barrier by using the previously explained mechanisms to treat different brain-related disorders. And different nanoparticles and mechanism of to respond neuroinflammation. Once the brain encountered with different neurodegenerative diseases, the pro-inflammatory cytokines were released, which in response exaggerate neuroinflammation. And this neuroinflammation was treated by using different type of nanoparticles like liposomes, which are made up of single or multiple phospholipid bilayers and resembles with our cell membrane. And the polymeric nanoparticles are solid colloidal dispersion of biodegradable and biocompatible polymers, such as polycyanoacrylate, polyester such as polylactide, PLA, PLG, and several other natural proteins and polysaccharides are used to formulate polymeric nanoparticles having the size range of 10 to 100 nanometers. Nanoemulsions are oil in water, water in oil. Emulsions are colloidal particulate systems that are composed of edible oil. Surface activation means surfactants, and these surfactants are used in fluidization of cell membrane and increase the permeability of these nanoemulsions, which permeate across the blood-brain barrier and deliver drug in the brain site. These are having the range of 100 to 500 nanometers. The solid-lipid nanoparticles is also an attractive tool to deliver drugs across the cell, across the blood-brain barrier in the brain. These are made up of triglycerides, fatty acids, steroids, cholesterols, and vexes. Dendrimers are also an attractive polymeric structures having 3D symmetrical architectures having different projections known as generations, which possess different functional groups on the terminal surfaces that can be easily functionalized with different ligands. Microneedles and brain-related disorders. Once nanoparticles or to tackle the different drug-related problems was formulated, means to avoid the size issue, to avoid the lipophilic, hydrophilic nature of drugs, we utilize nanotechnology. But once nanoparticles are formulated, we used to deliver these nanoparticles by using a suitable route at the brain site. First, oral route is not a feasible route to deliver drugs inside the brain because in oral route we encounter first pass effect, large transit time to reach the drugs at brain site. And the other modern technique used to deliver drugs in the brain site is microinjections. But current microinjections methods using hypodermic needles, which suffers large damage and irritability to infuse small amount of drugs. The smallest hypodermic needles that are commercially available 34 gauge with OD of 184 micrometer for neuro application, but having an extra cost. Thus, we need a specific route to deliver these nanoparticles across the blood-brain barriers. And we can utilize an emerging microneedle technique to deliver our drugs at a specific site by avoiding a pain and invasive injections, hypodermic needles. These are user friendly, means patient friendly. Patient compliance is the major reason to use these micro needles. They are small enough, a layman can easily be used these micro needles by a single press. The microneedles are far better than these hypodermic needles, which involves long syringes, which is very a painful procedure for every patient and needs a practical or experienced person to deliver hypodermic needles. And other local or topical transdermal delivery systems are gels, paste, hydrogels, but these delivery systems are unable to deliver optimum amount of or all amount of active moieties loaded in these particular systems. These particular system is used permission enhancers to open the pores of skin to deliver the drugs or nanoparticles across the stratum corneum. But these are unable to deliver all drugs because they are vanished by having a large aqueous amount in these gels, hydrogels. But microneedles have least aqueous system and is the best delivery method to deliver the drugs across the stratum corneum and penetrate directly in the stratum corneum and deliver drugs. Mainly, we utilized 7 different types of microneedles such as solid microneedles, coated microneedles, hollow microneedles, soluble, degradable microneedles, swellable, and porous microneedles. These microneedles utilize the mechanism can be utilized the mechanism of the drug to deliver at brain by using macrophages and monocytes. Once there is inflammation in the body, in brain, macrophages and monocytes activated in the blood circulation and move towards the site of inflammations. Thus, these macrophages and monocytes can be utilized to deliver our nanoparticles exactly at the brain sites of brain inflammation. And these microneedles can be used to deliver our nano drug by the help of macrophages at the brain infected cells. But the challenges faced by the nanoparticles based microneedles to deliver drug effectively and safely brain targeted delivery systems are these nanoparticles should be biodegradable and in nature and provide effective and safe amount. A non-invasive technique was utilized to avoid patient compliance, which is encountered in IV and other invasive routes. A eco-friendly green approach should be developed for the preparation of nanoformulations. The factors like shape, size, charge, and moiety attached to nanoparticles should be well elucidated and evaluated, which is essential for the crossing and developing brain-targeted delivery systems. These are the references which I utilized in my presentation. Thank you.
Thank you, Ikra Chaudhary, for such a productive presentation. Our third presenter of this session is Aisha Arouj from Kohat University of Science and Technology. She is going to present her research work on comparative analysis of Eucalyptus globulus, Calotropis procera, and Azadirachta indica against larvae of Aedes aegypti. Please, Aisha Arouj from Kohat University of Science and Technology.
Bismillahirrahmanirrahim. I am Aisha Uruj, Department of Zoology from Kohat University of Science and Technology. Today, I will present a topic entitled Comparative Analysis of Eucalyptus globulus, Calotropis procera, and Azadirachta indica against larvae of Aedes aegypti. The contents of my presentation will be introduction, objectives, material and methods, results, conclusion, and recommendations. First of all, I will introduce my topic. Aedes aegypti belongs to order Diptera, family Colicidea. It is a very important vector of many diseases such as dengue fever, chikungunya, yellow fever, and the Zika virus. Dengue fever is the most challenging vector-borne disease worldwide, mostly in tropical and subtropical regions due to the huge number of cases every year. Currently, the most important steps in preventing this disease are mosquito control and personal protection against mosquito bites. In current study, 3 plants were selected. One is Eucalyptus globulus, belongs to family Myrtaceae, also known as blue gum. Second one is Calotropis procera, belongs to Asclepiadaceae family, also known as ox. And third one is Azadirachta indica, belongs to family Meliaceae, commonly known as neem. Objectives. To assess the in vitro larvicidal activity of Eucalyptus globulus, Calotropis procera, and Azadirachta indica. And the second one is to compare the larvicidal activity of the selected medicinal plants against the larvae of Aedes aegypti. Material and methods. First step is sample collection, then grinding, and then preparation of crude ethanolic extract. After that, collection and identification of mosquito larvae, then larvicidal bioassay, and the last step is statistical analysis. Results. This graph shows the larvicidal effect of Eucalyptus globulus against Aedes aegypti larvae. The horizontal line shows concentrations and the vertical line shows mortality percentage. Eucalyptus globulus shows 33% mortality at 5 mg per ml and 37.22% at 10 mg per ml and 50% at 20 mg per ml and 57.22% at 40 mg per ml. This graph shows the larvicidal effects of Calotropis procera against Aedes aegypti larvae. Calotropis procera shows 43% mortality at 5 mg per ml, 49.44% at 10 mg per ml, and 56.67% at 20 mg per ml, and 70% at 40 mg per ml. This graph shows the larvicidal effect of Azadirachta indica against Aedes aegypti larvae. Azadirachta indica shows 40% mortality at 5 mg per ml, 43.33% at 10 mg per ml, 50.56% at 20 mg per ml, and 61.67% at 40 mg per ml. Conclusion. The overall mortality was higher at 40 mg per ml and minimum was at 5 mg per ml. All the 3 plants are effective against these vectors, but the Calotropis procera shows high mortality. Recommendation. The chemical characterization may be found out of ethanolic extract of Eucalyptus globulus, Calotropis procera, and Azadirachta indica up to compound. Thank you.
Thank you, Aisha Aruj, for delivering such a delightful presentation. Our fourth presenter is Noor Fatma from University of Agriculture, Faisalabad. She's going to present her research work on evaluation of the antibacterial activity of various plant extracts against clinical bacterial strains. Please, Noor Fatma from University of Agriculture, Faisalabad.
Bismillahirrahmanirrahim. Assalamu alaikum. Good morning, everyone. I'm Noor Fatma, PhD research scholar at University of Agriculture, Faisalabad. I'm very thankful to organizer and convener of conference for providing me the opportunity to present my research work on evaluation of antibacterial activity of various plant extract against clinical bacterial strains. Today, all over the world, plants are used for preparation of medicine due to the emergence of antimicrobial resistance. Recently, WHO estimated that 80% of people worldwide rely on herbal medicine. There are two types of phytochemical, primary metabolites and secondary metabolites. Primary are essential for growth and metabolism, while secondary is for defense mechanism. For example, inhibition of cell wall, nucleic acid and protein synthesis, and disruption of cellular membrane in their metabolism and capsule formation, while they also suppress quorum senses, formation of biofilm can also be controlled. The two most potent perennial plants, Calendula and Centella asiatica, were used for this research. Both plants are used for the treatment of anemia, fever, skin, and wound infections. Objective of the research is to identify the occurrence of Staph aureus, VRSA, E. coli, and Pseudomonas aeruginosa from clinical samples. Antibacterial profiling of ethanol and methanol extract of Centella asiatica and Calendula against clinical isolates. The combined effect of Centella asiatica and Calendula plant extract with commercially available antibiotic tests. To assess the clinical samples for the presence of Staph aureus, VRSA, E. coli, and Pseudomonas, 19 different wound, pus, and abscesses samples were collected from Allied Hospital, Faisalabad. These isolates were further purified on selective media and were identified by Burgess Manual of Determinative Bacteriology by performing a series of biochemical testing. After the preparation of plant extract of Calendula and Centella asiatica in ethanol and methanol extract, antibacterial susceptibility testing was performed by this Stepherian method. To examine the resistance among bacteria, antibiotic susceptibility testing using amikacin, gentamicin, ciprofloxacin, and ceftazidime. The synergistic effect was performed on antibiotic-resistant bacterial strain to identify the combined effect of plant extract and antibiotic test. To investigate the lowest amount of plant extract required to inhibit the bacterial growth, minimum inhibitory concentration was performed using 96-well microtiter plate, and to observe the lowest quantity of plant extract used to kill the bacteria, the well showing the MYC would further subculture on nutrient media to determine their bactericidal concentration. All of the findings show that Staph aureus, VRSA, E. coli, and Pseudomonas were frequently present in wound, pus, and abscesses sample. Antibacterial activity of plant extract indicate that Staph aureus show the highest activity against Centella asiatica, methanol, and ethanol extract, while VRSS show a highest activity against Calendula methanol, while lowest against Calendula ethanol extract. E. coli and Pseudomonas aeruginosa show the highest activity against Calendula ethanol extract, while lowest against Centella asiatica methanol extract. Antibiotic susceptibility results show that Pseudomonas aeruginosa and E. coli show 100% resistance towards ciprofloxacin, and Staph aureus was 20% resistant to ciprofloxacin, ceftazidime, and amikacin. VRSA show 100% resistant toward ciprofloxacin and ceftazidime. For synergistic effect of antibiotic and plant extract, antibiotic-resistant strains were selected to observe the combined effect of antibiotic disc and plant extract. The synergistic impact was observed by combining antibiotic and plant extract, and zone showing a higher zone of inhibition was observed as compared to their individual effect. For example, as you can see, Pseudomonas show 100% resistance towards gentamicin, ciprofloxacin, and ceftazidime, but after synergism show a zone range of 8 to 20 millimeter. Minimum inhibitory concentration was observed with increasing concentration of bacteria, for example 70, 80, and 90 microliter, and decreasing concentration of plant extract 30, 20, and 10 microliters. So by increasing the concentration of plant extract, the number of bacteria decreased as determined by MIC. All of the plant had MIC at 70 plus 30 microliter concentration against Staph aureus. Minimum bactericidal concentration was also determined, in which Centella asiatica, ethanol, and methanol extracts show MBC against Staph aureus at 30 microliter and 70 microliter concentration of MIC, and minimum bactericidal concentration was achieved at lower concentration of Centella asiatica methanol extract against E. coli, while higher concentration of Centella asiatica ethanol extract and methanol extract was required for Staph aureus to show a bactericidal concentration. All of the plant extracts show zone of inhibition against clinical isolates. Ethanol extracts, among them, ethanol extract of calendula show highest zone of inhibition among isolates. By combining the antibiotic and plant extract, greater zone of inhibition was observed as compared to their individual effect. By increasing the concentration of plant extract, the number of bacteria decrease, so these plants can be used as an active ingredient for the treatment of wound and various infections. Thank you.
Our fifth presenter is Aisha Nawaz from University of Agriculture, Faisalabad. She is going to present her research work under the title of Evaluation of Antimicrobial Activity of Lychee chinensis peel extract against various foodborne pathogens. Please, Aisha Nawaz from University of Agriculture, Faisalabad.
Assalamualaikum. My name is Aisha Nawaz. I'm doing my PhD from Institute of Microbiology, University of Agriculture, Faisalabad. The topic of my presentation is evaluation of antimicrobial activity of Lychee chinensis peel extract against various foodborne pathogens. Introduction. Food safety is a leading concern for public health worldwide, and food pathogens remain the major factor as the causative agent for foodborne diseases. Food spoilage due to the microbial contamination and oxidation of nutrients are the main reasons. The oxidation of nutrients occur because of the usage of the different preservation techniques that has required from the heat, temperature, or any kind of chemicals. And foodborne diseases are responsible for nearly 600 million illnesses and 4,20,000 deaths occur annually because of these foodborne diseases. Food and Agriculture Organization has reported that as economic loss of 1.3 billion tons of food products occur annually, and several preservation strategies to control microbial spoilage, such as chilling, reduction of water activity, acidification, pasteurization, aeration, chemical antimicrobials has been used in the past, but they have adverse various limitations like acquisition of the microbial resistance, adverse health issues, apart from that, environmental pollution, and above all this is the oxidation of the nutrients of the food because of the temperature, one of the things. For that reason, natural, cheap, and safe preservatives like plant antimicrobials can be used. Lychee chinensis is an exotic flu belonging to the Sapendaceae family, having 138 1 genera. Lychee chinensis grow in both tropical and subtropical regions and indigenous to Vietnam, China, Philippines, and contain various metabolites like flavonoids, phenolic content like procyanidin, B4, BTU, and epiphytogen, and they are rich in phosphorus, iron, vitamin A, B, and C. They have strong antiviral, antithrombotic, and antioxidant and activities. from the nutritional point of view. Although they have in vitro activity of the lichanesis leaves and seed have been reported, but the in vitro activity of the wheel and the in situ activity is not reported yet. So for that reason, this topic is choose. The objective of the study was to isolate and identify different foodborne microbes such as Staphylococcus aureus, Bacillus cereus, Escherichia coli, Pseudomonas aeruginosa, Salmonella enteritis, Aspergillus niger, and Candida albicans from various food samples to evaluate the antimicrobial activity of lichanesis, bale, seeds, and leaf extract against foodborne pathogens both in situ and in vitro. First of all, collection of plant material, preparation of peel, seeds, and leaf extract, then isolation and identification of foodborne pathogens. Then in vitro diffusion assay will be done, and in situ food matrices will be used like fish and chicken for the antimicrobial activity analysis. Next, collection and preparation of plant extract. For the collection and preparation for plant fruits, lychee fruit and leaves will be taken from the different gardens, and then washed and peeled off and dried. And after drying process, it will be grinded into milk powder form, and then with the help of 85% ethanol in it, ethanol, the 85% ethanolic extract of the leaves, seed, and peel extract was made by using conventional method and oxygenated method. And then isolation, identification, and in vitro antimicrobial analysis. Sample collection, first of all, is the main step. For different food samples were taken, and then they were grown over selective agar. The growth then growth appears on selective agar after seeing the microscopic characteristics of the bacteria and fungi. They were then gram staining was done for the bacteria, and for fungi like the phenol cotton blue stain was used. Then after microscopy, biochemical tests was used for different biochemical tests like methyl red, indole rest, straight test, test, catalase test was used to see which bacteria give positive test or negative test. Then antimicrobial susceptibility test was done by diffusion method in vitro against all these plant extracts. Then bacterial strains showed that like Salmonella enteritis was grown on Shigella agar strain, gram negative, catalase positive test, methyl red positive test, citrate positive, and motile positive. In the similar way, all these bacteria show these characteristics as shown in the table. Antibacterial susceptibility was seen against both conventional extract prepared both against the conventional method and software repeated method, and as a control test, ciprofloxacin was used. As antibacterial susceptibility testing seen against the leaf extract showed maximum growth against Salmonella enteritis was and Salmonella aeruginosa. While peel extract showed maximum against Staphylococcus aureus and E. coli. While seed extract showed maximum growth inhibition against E. coli and Salmonella enteritis. So, as the Salmonella enteritis and having some staph aureus are important, so they have been used in situ activity was checked against them also. So inhibition zone, again diameter of the extracted leaves and Chinese peel extract. So these Aspergillus niger and Candida albicans were the two fungi against which antimicrobial activity of the Chinese peel extract was seen. And from the graph, you can see that they show the ERPP indicated the extracted lychee peel powder that showed the growth at different days of the bacteria fungi, while the control is something having no control having negative control, having no antifungal in it. So the results and shows that the Chinensis peel extract has inhibitory effect against Candida albicans much more than the Aspergillus niger. Even the Candida albicans were grown for the 14 days. In situ antimicrobial activity was done. For the in situ activity, samples were taken and mixed with the inoculum and extracted and placed in the refrigerator for as a preservative and for a few days. And at different days, the samples were taken and then blended with the peptone water. And after severe dilution was poured over the general medium, selecting media for the bacteria and to see the colony counting growth with over that. And the results indicate this in this form that Streptococcus aureus has shown the minimum inhibition, has shown growth reduction in 4.94 CFU to 4.7 CFU, and while control have 5.6 CFU at the initial time, and they at the 14th day it has 7.3 CFU of the Staph aureus of the fish at the control, while the Salmonella enteritis fish showed that 3.1 at the initial time and 2.8 CFU at the 14th day. So, similar with the case with the meat, they have shown that the 3-point reduction of CFU from 3.8 to 3.4, while the control have also reduced the growth reduction, but that is because of the temperature and other factors. So, from this, it can be realized that ethanolic leishmaniasis leaves and seed extract showed antimicrobial activity against all selected food-borne pathogens. The strong inhibition was seen against Staphylococcus aureus, E. coli, and enteritis. And apart from them, the Candida albicans was also against Candida albicans was also seen. This Chinensis peel extract showed a growth reduction against Staphylococcus aureus and inhibit at not much that level, but it inhibited the growth of Staphylococcus enteritis growth on food matrices also, and can be used as a natural preservative agent for the preservation of the food. Thank you.
Thank you, Aishanwaz, for such an amazing presentation. Our next presenter is Mohammad Kabir from Thal University of Bakar. The title of his presentation is Causes, Impacts, and Solution of Rapidly Increasing Environmental Pollution in Pakistan. Please, Mohammad Kabir from Thal University of Bakar.
Bismillah ar-Rahman ar-Rahim. Respected chair, co-chair and audience, Assalamu alaikum. I am Dr. Mohammad Kabir, serving as an assistant professor and in charge at Department of Biological Sciences, Thal University Bakar, University of Sargodha, ex-sub-campus Bakar. First of all, I pay my thanks to the organizing committee of International Conference IBBC-2023 and Department of Physiology, Government College, University Faisalabad, who give me a chance of oral presentation in this an international conference. Audience, today the topic of my presentation is Causes, Impacts and Solution of Rapidly Increasing Environmental Pollution in Pakistan, a review. I am not going to be presenting any research paper. I just want to highlight an important issue of the environment through a review. Presentation layout includes the problem, that is the pollution, and its solution, that is the plantation. As we know that research starts with the problem and ends with the solution. Causes and effects of environmental pollution, importance of tree for sustainable habitat, pollution, pollutant type and issue, plantation campaign, solution for pollution, environmental calendar, conclusion and recommendation. certificates and publication of 2022 and 23. Environmental pollution. In this slide, we will see the causes, effect, and its solution. The major causes of environmental pollution are the overpopulation, urbanization, industrialization, transportation, deforestation, waste production, resource depletion, lack of education, and so on. And the effects include the biodiversity loss, floods, landslides, soil erosion, endangerment of species, natural disaster, global warming, climate change, and so on. But the lower level solutions include the control of population explosion, stop deforestation, promote reforestation, fine for illegal dumping, strict government regulation, reverse nature, reduce consumption level, and so on. A muscosol, an environmental expert, say that a tree that lives for 50 years generates a US dollar worth of oxygen, recycles huge quantity of US dollar worth of soil fertility, facilitates US dollar worth of the soil erosion, provides a huge quantity of US dollar worth of shelter for insect, birds, and animals. Besides, it provides flower, fruit, and seed. Our net loss is worth 57,381 US dollar when one tree falls are affected by environmental pollution. So the humble request to all the humanity on the surface of earth is that think before you cut a tree. What is the pollution, pollutant, their types, and the issue? You can see in the picture that it is our common practice that most of the industrial wastes are dumped outside the industrial wall or their boundary wall, and then by burning process different types of pollutants are being discharged into all components of the environment, including air, water, and soil. So what is the pollution? An undesirable change in physical, chemical, or biological characteristics of air, water, and soil that may harmful effects to the living organism is known as the pollution. Pollutant. The substances which cause the pollution are known as the pollutant, or simply we can say the residue of things we make, use, and throw away into the environment are known as the pollutant. Different types of pollutions include the air pollution, water pollution, land or soil pollution, noise pollution, thermal pollution, chemical pollution, and so on. What is the issue? We use the resources extensively and then are not responsible for the consequences. In the next slide, you can see that it is our common practice that we cut the tree by process of deforestation. One sin is the cutting of tree and another is their burning. By burning process, we are releasing different types of pollutant into the air as well as into the soil. In the second part of slide, you can see the anthropogenic activity, that is the treatment of the water, but such activities are not being taking place on the large scale in our environment. Here in the figures, you can see that how different types of the greenhouse gases are being produced into the environment. These are also the major pollutants of the environment, and these pollutants are mainly being discharged due to the industrial activity, transportation, construction purposes, waste production, road constructions, and so on. So what are the solutions for pollution? Here we will see the solution for pollution regarding to the some codes. That is, plants act as a natural lungs. Plant a tree and get oxygen for free. Actually, the only things which is giving existence to all living organisms on the surface of earth are the plants, because they give off the oxygen which is necessary for existence of all the living organisms. They are the producer of the food chain, and they can provide food to all the living organisms by the process of the photosynthesis. And by this process, plants are considered as the natural purifier of the environment because they take up the different greenhouse gases during the process of the photosynthesis, and most common gas, carbon dioxide, is being taken in the process of photosynthesis by the plants. It is our duty to save the environment's beauty. Let us go green to get our globe clean. In shortcut, we can say that green revolution is the best solution to arrest the environmental pollution in changing climate of the world. Here you can see in this slide that in our university campus, different entrance gates are also made by different types of the plants. We have planted a large number of plants in earthen pots in front of our academic building. We arrange different walks and talks in which students, faculty members, and staffs are involved in plantations activity. Our birthday director is planting a tree in front of the academic building. We also have collaboration with the district administration, deputy commissioner office, divisional, and district forest officer to carry out the plantations activity in our campus. We also collaborated with the Pakistan Army for plantations activity in our campus. As we know that clean and green environment is necessary for the existence of life and existence of the healthy brain. As we know that healthy brain can exist only in the healthy body, and healthy body or healthy species require a healthy environment. We normally celebrate on two important days in a year, that is our birthday and the Eid's day. But we must also know that environmental calendar have many important events in a year, such as the World Environmental Day is celebrated on the 5th of June, World Population Day on the 11th of July, Ozone Week from 16 to 23rd of September, and so on. The International Plantation Day is celebrated on the 18th of August. So what is the conclusion? If haphazard population growth and construction of new industrial structure, transportations and urbanizations activities go on without realizing the importance of trees, then probably there would be more environmental changes in near future. These environmental changes are harmfully affecting the habitat and health of the living organism. As I already mentioned that the clean and green environment is necessary for existence of organism on the surface of earth. If our environment is not clean, it means that we are not giving the sustainable environment to our next coming generation. So recommendation is that be a part of solution, not pollution. And the message, request, whatsoever you consider it, to all the participants is that each one must at least plant one. Here you can see in the slide that its heading is need to think. As in the first part of the slide, industrial revolution is discharging disturbance both in the land and in the air. And most of productive ecosystem, that is forest ecosystem, is also being destroyed due to the different anthropogenic activities. That is industrialization, urbanizations, and so on. You can see in the second right portions of the slide that without trees, the temperature is very high, and with trees, this temperature on the surface of earth is under control. Imagine if tree give off Wi-Fi signal, we would planting so many trees, we would probably save the environment. Too badly, they only produce the oxygen which we breathe and food which we eat. Request to all the humanity, to all the participants is that join the green revolution and stop the pollution. Here you can see in the slides some of the certificates which our University of Sargodha have obtained during its competitions in UI Green Matrix World University Ranking. During 2023, this position was 526, and in 2018 it was 712. And in the second portion of slides, you can see the Research Productivity Award during my PhD, which I have obtained from Pakistan Council of Science and Technology, Islamabad. These are the publication of 2022. I have published 10 papers of W and X categories, and during 2023, I have published 5 papers, out of which 4 belongs to W category and 1 belongs to the X category. It means that I am playing my role being as a inhabitants of this environments in the form of the publication. So save the environment to save yourself. Thank you. Be kind to your mind.
Thank you, Muhammad Kabir, for presenting your informative research work on IBBC 2023. The next presenter is Muhammad Iqbal, from Government College University, Faisalabad. He's going to present his research work on enhanced nickel removal with white willow from contaminated soil under the influence of PGPR. Please, Muhammad Iqbal from Government College University, Faisalabad.
Bismillahirrahmanirrahim. My name is Dr. Muhammad Iqbal, and the topic of my presentation today in this conference is enhanced nickel removal with white willow from contaminated soil under the influence of PGPR. So I am Associate Professor in the Department of Environmental Sciences and Engineering, Government College University, Faisalabad, Pakistan. Outlines. Introduction. Sources of soil pollution with nickel. There are different sources of soil pollution with nickel, effluents from different industries, industrial solid waste, coal fly ash, mining and smelting. So these are the different sources from where the soil is polluted with nickel. So hazards of nickel polluted soils to the environment: food contamination, they reduce crop yield, pollution of water bodies due to runoff, groundwater pollution, effect on soil fertility, effect on soil organisms. There are different remediation strategies for the remediation of nickel polluted soils. For example, soil excavation, soil replacement, solidification, vitrification, and washing. These treatments are harsh. They have more demerits and less benefits. They are costly, laborious, less acceptable by public, and they damage to the environment. Phytoextraction is a gentle remediation strategy that has more benefits and very less detrimental effects on the environment. Phytoextraction is the process of extracting heavy metals such as nickel, lead, cadmium, copper from the soil by different cumulative plants or high biomass producing plants. The positive effect of phytoextraction of the soil is increased fertility, increased water holding capacity, increased removal of metals from the soil, reduced metal from the soil, and decreased nickel toxicity to the plants. So what are the processes that happen during nickel phytoextractions? There are certain microbes that solubilize nickel in the soil, reduce nickel stress to the plants, enhance soil enzymes, and there are many different organic amendments that provide nutrients to the plants during phytoextraction and improve soil health. Problem statement. Nickel contaminated soil can contaminate groundwater, agricultural land, and water bodies. Thus, remediation of these soils is needed. Objective. The objective of our research is to investigate the role of rhizosphere acidifying plant growth promoting rhizobacteria for reducing soil pH exclusive in the rhizosphere of Salix alba, which is called white willow. The effect of reduced soil pH on the viability and phytoextraction of nickel from the soil. Biostimulating role of LM and AM. These are two organic amendments for enhancing the efficacy of nickel phytoextraction by Salix alba from nickel polluted soil formerly receiving nickel rich effluents from electroplating industry. Here LM is the organic compost prepared from green waste, and AM is the organic manure of cows, while the LM is the compost of kitchen waste. So the post experiment contained 6 treatments: control, B, LM, B plus LM, A, B plus AM. Here the B is bacteria that is going to acidify the rhizosphere of Salix. Nutrient sources are cow dung, which is called AM, kitchen compost LM. Pot size was 3 kg per pot size, the soil. Nickel contamination was 72 milligram per kg soil. Duration was 3 months. Plant name was Salix alba white willow. Materials and methods. Soil analysis. The soil analysis was conducted through standard protocols. For example, variable nickel in the soil was measured by DTP extract, pH with pH meter, cation exchange capacity by Rodas method. Soil texture was assessed by hydromether method. Soil enzymes, dehydrogenase, beta glucosidase, urease, acid phosphatase, and peroxidase were measured with the method explained in Birk et al. 2022. Bacterial count, microbial biomass, and organic carbon in the soil were done through various methods explained in Tokir et al. 2022. Plant analysis, nickel concentration and the content in shoots and roots was measured by using various standard protocols presented in Birk et al. 2022. Chlorophyll A and B in the plants were measured by using spectrophotometer. Results and discussion. Here we can see that soil pH. Soil pH, here we can see that the soil pH of the rhizosphere was relatively lower compared to the bulk soil. So rhizosphere is the zone that surrounds the plant roots. It's the most active zone where the microbes live and they perform their functions. In this figure 1, we can see that inoculation of bacteria has significant effect on reducing soil pH compared to control. So AM and LM, they did not change soil pH. But when LM plus bacteria was together applied in the soil, their effect was the highest for decreasing soil pH. Distribution of nickel in Salix plant parts, values of BCF, bioconcentration factor, and translocation factor of nickel, valuable nickel in the soil, and nickel removal from soil as factored by adding LM, AM, and Bacillus species. Here we can see that the highest concentration of nickel in shoots, roots, nickel contents in shoots, roots were the highest in B plus LM treatment. Similarly, bioconcentration factor, translocation factor of nickel removed from the soil, as well as TTB extractable nickel were the highest in the same treatment. Nickel distribution in plant parts and soil enzymes. After the nickel distribution in plant parts, we can see that the activities of soil enzymes, as well as the bacterial count, microbial biomass, and organic carbon, they all were the highest in bacteria plus LM treatment. Conclusions. Bacteria acidified the bulk and rhizosphere soil more than LM alone inoculum. The highest nickel solubility was observed in B plus LM treatment, leading to the greatest nickel concentrations, contents, and nickel removal values. The highest soil enzymatic activities, microbial and bacterial biomass, organic carbon were found in B plus LM treatment. The B plus LM treatment can efficiently clean soil from nickel pollution and improve soil enzymatic activities. Thank you very much for hearing my presentation.
Thank you, Muhammad Iqbal, for delivering such an amazing presentation. Our last presenter is Amna Nazish from Kohat University of Science and Technology. The title of her presentation is Evaluation of Bacteriocin-Producing Bacteria Associated with Beehives of District Karak Against Multiple Drug-Resistant Bacteria. Please, Amna Nazish from Kohat University of Science and Technology.
Assalamu alaikum. Very good morning to all the eminent dignitaries and research scholars present over here. Myself, Amna Nazish, a research scholar at Kohat University of Science and Technology. First of all, I have to thank my supervisor, Sarishtia, who guides me throughout my research work. And I would also like to express my gratitude towards the organizer and conveners of this conference for providing me the opportunity to present my research work in front of you all. The title of my research work is Evaluation of bacteriocin producing bacteria associated with beehives of District Kirk against multiple drug resistant bacteria. Bacteriocin producing bacteria. First, we will discuss bacteriocin. These are the proteins. The word also clue about something related to bacteria. So, bacteriocin, these are the proteins which are produced by bacteria. Both type of bacteria, gram-positive and gram-negative, produce such proteins, but the majority of bacteriocins are produced by gram-positive bacteria. Content, introduction, problem statement, objectives, material and method, results, conclusion, recommendations. Introduction. Honey beehives harbor a diverse community of bacteria, some of which are known to produce bacteriocins. Bacteriocins are antimicrobial peptides produced by bacteria that can selectively kill or inhibit the growth of other bacteria. For example, if some bacteria like gram-positive bacteria produce some bacteriocin, they can inhibit the growth of gram-negative bacteria. So in the same case, gram-negative bacteria, the bacteriocin of gram-negative bacteria will work on against the gram-positive bacteria. Antimicrobial resistance is a significant challenge for the healthcare system, and finding effective strategies to combat that is crucial. Bacteriocins are one potential option for combating drug-resistant bacteria. They have several advantages over conventional antibiotics, including their ability to target specific bacterial species. Usually, we use antibiotics against bacterial infection, but in the recent report of World Health Organization, they talk about these antibiotics have side effects, and the recent one of their recent report about 2023 about how these antibiotics affect the gut microbiota of human beings. The gut microbiota, which is a very beneficial for human health, they have a side effects on such microbiota. So we have a option to substitute the antibiotics in the form of bacteriocin. Objectives: to investigate the bacteriocin producing bacteria present in honeycombs of different species and District Kirk especially, to observe the effect of bacteriocin producing bacteria against multiple drug resistant bacteria. Problem statement: the emergence of antimicrobial resistant results and morbidities and mortalities associated with many bacterial infections and as a major concerning issues and front of healthcare system. Material and methods: collection of sample. First we taken a sample, 8 samples of honeycombs and from different apiaries and District Kirk, and these samples were honeycomb were then crushed, homogenized and normal saline, and their solution were well then cultured on a beach-high agar media. Beach-high agar media, beach-high as the abbreviation of brain heart infusion agar. This is a media, this is an agar which is non-selective. It means different types of bacteria can grow on such media. But there are some also a selective agar which are specific for some bacteria. But beach-high agar is a non-selective. There are a lot of bacteria can grow on them, like nutrient agar. And then gram staining, biochemical test, and we get a result of 6 samples were positive for Bacillus subtilis and 2 were negative. It shows that the honeycomb of district Karak as have a very high quantity of Bacillus subtilis. Presence of it, it gives a clue, an average value that is 6 samples were positive for Bacillus. It means there is a high existence of Bacillus subtilis and honeycombs of district Karak. And then for detection of antibacterial activity, we use 2 types of test, agar spot test and then agar well diffusion assay. At the result of these 2 methods we used, we get results like Bacillus subtilis, which is a bacteriocene producer, show an inhibition zone about 20 mm against E. coli, which is a multiple drug-resistant bacteria, and 18 mm against Streptococcus mutans. Conclusion. Finding of the study supported the presence of bacteriocene-producing bacteria, Bacillus subtilis, and majority of honeycomb of district Karak, and also such bacteria show antibacterial response against MDR bacteria like E. coli and Streptococcus mutans. Both E. coli and Streptococcus mutans are multiple drug-resistant bacteria. Streptococcus mutans, which is the major cause of tooth decay, related to tooth decay diseases, different types of infections of tooths and jaws. Recommendation. It is recommended from the study that although we explore the bacteriocene-producing bacteria, but for enhanced bacteriocene activity, the development of proper medium and condition needs further study. So this is all about my research work. Thank you so much.
Thank you, Amna Nazish, for such a productive presentation. I would like to thank everyone for your participation on our first International Brain and Biomedicine Conference, IBBC 2023. Our next moderator will continue the next session. Thank you for being attentive. Stay tuned. Allah Hafiz.